Good afternoon, sir. Good afternoon, everyone.
Good afternoon.
I think I should start the session. It's already 6 past 3.
Yes, ma'am.
Okay, so today I'm going to talk about monoclonal antibody as drug candidate. And I will start with my introduction. I'm Manisha Pritam. I'm a project scientist at AIMS Gorakhpur. Okay, so let's start with the introduction. The first monoclonal antibody that was approved by United States FDA, it was anti-CD3, OKT3. It's also known as muromonab CD3. CD3. It's an antibody that binds to CD3 protein. In 1986, it was first approved by FDA. Till 2022, according to the literature data, around 162 antibody therapies have been approved, but this is only for American approval. There are some European approval also. So in approval, there is only 100 or 150 antibodies that are approved. Not much antibodies are approved by European countries. So if we talk about FDA, it's only 162 antibodies that are approved. And these approved antibodies are targeting wide range of diseases that include cancer, immune-related disease, infectious disease, and some hematological disease. For example, ACT-EMARA, it's also known as tocilizumab. This antibody was used for COVID. So if we talk about the global therapeutic So if we talk about the global therapeutic monoclonal antibody market, it was approximately $115 million in 2018, and it was expected to generate a revenue of 150 billion by the end of 2019. And it was estimated that at the end of 2025, it could be 300 billion. Now it's 2026, so new data still need to be published. So these all data are from till 2025. The market of therapeutic antibody drugs has experienced explosive growth as new drugs has been approved for treating various human diseases, including many cancer, autoimmune, metabolic, and infectious diseases. Monoclonal antibodies are man-made proteins that act like human antibodies in the immune system. There are 4 different ways they can be made and are named based on what they are made of. So if it's made from murine, or you can say mouse protein, we name it OMAB. If any antibody names end with OMAB, this means this antibody is made from mouse protein. If the antibody or the antibody protein prepared by the part of mouse and a part of human, then we call it ZMAB. At the end of the antibody name, there should be ZMAB. So why it's chimeric? Because in this chimeric antibody, there should be some part of mouse and some part from human. Then there is third type, it's humanized. So there's some small part of mouse protein attached to human proteins. In this case, we name it ZUMAB. So at the end of the name of that antibody, we add ZUMAB. The last type is human. So in this type, the human protein is fully antibody is prepared from full human protein. The whole sequence is from human. So it's known as ZUMAB. Whatever the name of antibody, if it's end with ZUMAB, this means this antibody is a fully human protein. There is no mouse protein. Now, there are 2 different categories of monoclonal antibody therapies. One is naked monoclonal and second is conjugated monoclonal antibody. So in naked monoclonal antibody therapies, we just use antibodies and there is no drugs or any radioactive material that is attached to that antibody. We just use the human or mouse antibody. There is no drugs. While in case of conjugated monoclonal antibodies, what we do, we use some kind of linkers that binds drugs to the monoclonal antibody. So in this case, what happens? Suppose you are trying some targeted therapy for drug and you want your drug to go and bind to some specific cell or some specific location. For example, in case of cancer, there are certain proteins that express on the surface of only cancerous cell. In that case, what we want to do, We just want to kill the cancerous cells, but not the human cells, the normal cells. So in that case, what we can do, we can bind our drugs to those specific antibodies that are specifically for cancer. And after conjugation, when we put this antibody inside the cancer patient, what happens? These antibodies specifically go and bind to the cancer cells. So this is also a very good approach. What are the applications of monoclonal antibodies? Okay, so I hope all of you are familiar with monoclonal antibodies and polyclonal antibodies. So normally in nature, there is polyclonal antibodies, but humans are generating some artificial antibodies, so we name it monoclonal antibodies. What are their differences? We will discuss later. So here, what we can use for, like, what are the application of monoclonal antibodies? This can be used to prevent infectious disease during disease season. For example, malaria infection is on peak during rainy season. So what we can do, we can just take monoclonal shots or monoclonal antibody therapies during season, because most of the time antibodies deplete. So it's most of the time it takes like 6 months, and after 6 months your antibodies deplete. So if there is a season of a specific disease like flu is also a seasonal. Like in winter season, there is always we hear about some, okay, there is some flu is ongoing. So before winter, we can take monoclonal antibody. It will prevent us from those kind of flus. Next is it can also protect travelers. For example, you are going to some South African region where there are some specific diseases. For example, malaria. So in that case, you can take monoclonal antibodies. It will prevent you from infection. And this is very good approach for pregnant women and small children's like infants or children's. And it also helps eliminate disease from defined geographical areas. Besides this, monoclonal antibody can be used as therapeutics during infection. For example, if you get infected from any kind of pathogen, you can take these monoclonal antibody and it will help you or it will protect you from that specific pathogen. For example, if you are infected with COVID-19 or SARS-CoV-2, if you're infected with SARS-CoV-2, you can take the monoclonal antibody and it will help you to reduce the effect of that pathogen and it will help you, it will protect you from that SARS-CoV-2. Most of the time, people generate antibodies, but if there is some immunocompromised person, we can use monoclonal antibodies because most of the time, elderly people or some people who have chronic disease or they are suffering from HIV or some other kind of diseases, there is possibility that they are immunocompromised. In that case, we can also use monoclonal antibodies. And these monoclonal antibodies can also use for drug delivery. So drug delivery, for how we use drug delivery, we use conjugated drugs and antibodies therapy. And it's specific, like mostly widely used for cancer, but you can also use for some other diseases. So if you are interested in working on monoclonal antibody, you should explore FDA approved monoclonal antibody list. Here I'm showing just a small data because it's not possible to show all the data here. So in table 1, you can see the monoclonal antibody, the name, the first column, it's showing the name of that monoclonal antibody. So you see there is some ONAB, ZMAB, UMAB. There are some different ending. Like if you see at the end, ZMAB, can anyone see here? ZMAB means it's chimeric antibody. And if you see OMAB, this means it's mouse protein. And if you see UMAB, this means human protein. So here you see it's OMAB. So this means this is from mouse. This contains mouse protein. And here you can see murine IgG2. So they are providing all the data here on this FDA portal. They will also give you the brand name, like which brand is producing this specific monoclonal antibody. You can also see the company name, what are the targets. For example, Muromonab CD3. This is the target is CD3. This means this antibody is going to bind with CD3. And if you see what kind of technology they are using hybridoma technology. Some people are also using phase display. So it's a very popular trend, like phase display is also very popular. So some people are producing antibodies using this phase display method. And what are the purpose? So, so what are the purpose of this monoclonal antibody? You can also see, like we use this CD3 antibody for kidney transplant rejection, and it's approved in 1986. You can also see in which year which monoclonal antibody is approved and what are the purpose. So if you see here, there is trastuzumab. This is a human protein. UMAB means human protein. Okay. And the target is HER2 receptor. So what happens when this monoclonal antibody bind to HER2 protein, it helps to kill breast cancer cells. Similarly, for asthma or for colorectal cancer or some other diseases, you can use these kind of monoclonal antibodies. When you go to this FDA portal, you can easily read more details of each monoclonal antibodies. So let's start with some introduction part of this B cell activation, how it works. What happens whenever there is some pathogenic bacterial cell, it enters inside our body. There are some antigen presenting cells, or we can say the B cells. These B cells have B cell receptors, and these B cell receptors interact with this pathogenic bacterial cell. This is a direct approach. So we say this is a T cell independent B cell activation. So what happens whenever this bacterial cell or any antigenic particle interacts with B cell receptor, these B cells activates and produces antibody. The second approach, what happens when there is T cell activation? So what happens, there is some other mechanism ongoing for MHC class 1 or MHC class 2. So if in case of MHC class 2, CD4 T cell activates and it produces some cytokines that activates B cell, and that B cell start producing some antibodies. So the cells, those are producing antibodies, we call them plasma cells. And there are some cells that differentiate as memory B cells. So when there is some second encounter of pathogen, these memory B cells become active and they produce antibodies. So this is very simple phenomena that B cells get activated. It could be T cell independent or it could be T cell dependent. So based on this activation, we classify T cell in 4 different stages. First is naive stage. Naive means there is no coding or no activation of any kind of antigen on this B cell. Once this naive B cell interacts with any kind of antigen or pathogen, it activates. So during activation, this B cell 1 changes into B cell 2. This activated B cell can produce or differentiate into plasma cell or memory B cell. So plasma cell here we call it B cell 3, and plasma cell, some of the cell like these activated B cells, some of the B cells differentiate into memory B cell. So we call it stage 4 B cell. These memory cells used by our body when there is second encounter. If I'm too fast, you can tell me. I will be slow. So now, how many types of antibodies? So here in this picture, you can see this is heavy chain, this is light chain, and this upper part of this immunoglobulin, these are known as variable region, because most of the time these regions are highly variable, and this region are most of the time constant region. So this is the structure of an antibody. So in humans, there are 5 different type of antibodies known as IgM, IgG, IgA, IgD, and IgE. So whenever there is an infection, the first antibody response from our body, IgM. Always remember, this IgM is going to activate first. Now here in this table, if you can see, IgM is a pentamer, IgA is a dimer, while IgG, E, and D, these three are homomer. And they have different heavy chain, the number of antigen binding sites, like for IgM, they have 10, for IgG, 2. So here you see the variable region. Because they have 2 variable region, they have 2 antigen binding sites. That's why if it's monomer, they have 2 binding sites, and if it's trimer, it becomes 4, and if it's pentamer, it becomes 10. And based on their molecular weight, you can see how many molecular weights are here, the percentage in our body. So you see the IgG is the highly available in our body, it's 80%. Most of the time we get infection, IgG are playing a major role. And again, the cross placenta IgG. This is also an important point, that which of the antibody can cross the placenta. Only IgG can cross placenta and go to the baby. So you see, all others are not able to, either IgG and D and E, both are homomer, still they are not able to cross that placenta. And you can also read the function. So here you see, if you got a primary antigen exposure, what happens? After 10 days, you can see an induction of IgM antibody. The first immunoglobulin is IgM. After few days, you see here, 15 days, at 15 days, there is increase of IgG immunoglobulin. So this is a primary response. Secondary response means when there is second time of exposure. What happens? Even at the secondary stage, you see the first uh, immunoglobulin is IgM that is inducing, and then IgG is inducing. So this is what happens whenever you got some infection. If you talk in the term of vaccine, what happens when you take the first dose, your IgG will not be very high. That's why we take booster dose. So during booster dose, what happens? It gives us a secondary antigen exposure. And because of that booster dose, we get a huge amount of IgG. So this is what happening during infection or during vaccination. I hope this is clear. Now, if we talk about antibodies, so there are 2 types of antibodies I told you in the starting of this session, that there is polyclonal, naturally there is polyclonal antibody, but humans are designed some monoclonal antibodies. So what is difference between polyclonal and monoclonal? So polyclonal is cheap because it's natural. We just have to extract. Monoclonal is expensive because we have to express, then we have to purify. So it's expensive. In polyclonal, it's a mixed population of antibodies. But in monoclonal, it's a homogeneous population of antibodies. So if you have any confusion, I make it clear that, for example, this is polyclonal antibody. So this antibody can bind to different antigens. You see? Okay, so this is antigen, and there are certain amino acid regions that is known as epitope. I hope you all are familiar with the epitope. I have discussed about the epitope in last session during vaccine development session. So the epitopes, you see, these are the region where epitopes are present. So these epitopes, suppose this is E1 epitope and this is E2 epitope. So what is happening in polyclonal? This antibody can bind to epitope 1 as well as epitope 2. But in case of monoclonal antibody, this antibody can only bind to epitope 1. It's not going to bind to epitope number 2 because they have different configurations. It's bind to different area of antigens. Okay, same thing. Tolerant to small change in protein structure. Okay, so here if there is some kind of changes happening in the antigen, for example, there is a strain. If you talk about SARS-CoV-2, there is protein spike protein. And we all know there is a Omicron variant. After that SARS-CoV-2, there is Omicron variant. So if there is any changes in Omicron variant, there is mutation in the protein, this polyclonal antibody can bind. But the limitation is that maybe there is possibility that monoclonal antibody will not be able to bind because it will not be able to identify and it increase the cross reactivity. So there is chances that this polyclonal antibody, if you are using for COVID-19 or SARS-CoV-2, it can also be used for Zika virus infection or some other pathogenic infection. So there are lots of literatures if you search. There are antibodies that is used for HIV, but they are also showing good reactivity against Zika virus or some other virus or any other pathogens. So this is the very good property of this polyclonal antibody. But in case of monoclonal antibody, it loses reactivity, like it cannot bind to some other antigen. So how we produce polyclonal antibody? Okay, so if you see here, most of the time when we perform ELISA, okay, so can anybody tell me all the students in this batch are computational or bioinformatics background or some have wet lab experiment background? Anyone there? They have molecular biology or biotechnology experimental background?
Yes, ma'am. Experimental background I have.
Okay, so I'm going to explain here. So if you are going to perform some kind of ELISA or Western blotting, you need some antibodies. In that case, what we do, we just purchase some antibodies. It could be rabbit antibody or it could be mouse antibody. So right now in clinical studies, we use rabbit monoclonal or polyclonal antibodies because it's very good. Like it's it's very good for clinical purpose. So what happens? We just take antigen, we infect this rabbit, and in this rabbit there is some B cell activations, and then the B cell start producing some polyclonal antibodies, and these polyclonal antibodies we can harvest from this rabbit. And this secreted antibodies by this rabbit then commercialized. So this is a very ancient approach, very common approach. If there is some animal house in your university or your research center, you can just go and explore this phenomena. Like there is only 5 to 6 weeks. It will take only 5 to 6 weeks to complete this whole process. Now talk about monoclonal antibody production. We all have studied about hybridoma technology. So this technique was introduced by Kohler and Wilson in 1975. This technology widely used for antibody production based on the fusion of mouse immunized B stem cells and myeloma cells. In this technology, we use some immortal B cells and then we clone and screen some monoclonal antibody producing antibody cells. So what happens here? We just take a antigen, whatever you are interested, we inject in mouse and we isolate some B cells and we use myeloma cells because they are immortals. Like if used here, they become hybridoma. So how we will know that okay, our fusion will successful or not? For that we use hat medium. So we use hat medium on ELISA plate. In this plate, when you grow, isolated B cells will die after 1 week, and this myeloma die because of this A. I will explain in next slide here. So what is happening here? This hat media contains hypoxanthine, aminopterin, and thymidine. So what happens? Aminopterin inhibits VHFR that blocks the de novo pathway of DNA synthesis, and ultimately cells cannot make purines or thymidine normally. And because of this, this myeloma cell dies. So you see, after 1 week, this B cell automatically die because it's not immortal. And because this is immortal, this also needs some uh, purines or thymidine for their synthesis. But if you inhibit this process by aminopterin that is present in hat media, this will also die. So what happens? Only hybridoma cell will survive because B cells have another pathway, like they have salvage pathway in their system. So what happens? In this media, there is hypoxanthine that convert into purine nucleotide by using salvage pathway. And because of this property, this hybridoma cell survive in this screening process. After this screening, we just expand these hybridoma cells. And after that, we purify these monoclonal antibody by different process. The next monoclonal antibody production is phage display. This was introduced in 1985 by Smith. It's also very popular nowadays. So the process refers to a gene sequencing coding for particular antibody that is integrated into DNA sequences of filamentous bacteriophage, which can be expressed on the surface of the bacteriophage capsule. The phage infects E. coli and then continues to display new phage in the host cell. If you are interested to learn more about this phage, just read this paper, Lim et al. It's very good paper if you are interested in phage display. So they expressed co-nucleoprotein, like it's a similar to SARS-CoV-2. So you can read for more details. I'm just showing some introduction for this phage display. What happens? We just select some B cell from infected people, like if some patients are infected with MERS-CoV or any other pathogenic, then we just isolate B cell and we use PCR for amplification of heavy chain and light chain of variable regions. And then we clone in phage mates. Then we transform these E. coli and then we prepare some library. What happens if there is no proper formation of this phage? It will not express antibodies on the surface. And if antibodies are not on the surface, they will not identify antigens. So what happens? We use ELISA plate. We coat our ELISA plate with antigens and we put our this phage display on this ELISA plate. And if there is antibody secretion on the surface or expression on the surface, it will bind to this plate. And when you wash, the non-binding antibody containing this specific phage display, it will be washed out. So what is the benefit? In this case, you can express multiple antibodies for different kind of antigens. For example, in case of COVID-19, there is envelope protein and spike protein. So if you want both antibodies for envelope protein and spike protein, you can just design two different phage here and you can express simultaneously. And once this phage is ready, you can just segregate them by using this simple screening protocol. After that, you can select, like you can segregate different phase for envelope protein and spike protein, and then you can transfect equalize for further production of antibody. Only one drawback is here that you can only express small fragments by using this phase display method. You cannot express the whole. So these are some example of FDA approved phase display drugs. That is, here you can see the brand name and the target like PDL1. These are used for cancer, like cancer treatment, and EGFR, it's also used for cell lung cancer. So most of them are developed for cancer, most of them. But there are some other like enough antibodies that is used for inflammation or rheumatoid arthritis or some chronic diseases like arthritis and psoriasis. So yes, there are lots of, like there is huge list that is FDA approved. There are huge list that are under clinical trial. So you can explore. If you are trying to design new antibody using bioinformatics approach, you should go and download the data for FDA approved and also clinical and preclinical trial, because most of the time there are common properties that is present in FDA approved and the antibodies that are in different clinical phase. So if you want to develop some model or you want to design new antibody, you should, excuse me, you should study about the properties of clinically approved, different, like in which phase that is present. Like there are lots of like thousands of monoclonal antibodies that are failed during different stage of clinical trial. Only few like 169 or 170 are approved by FDA, but that doesn't mean those antibodies are not good. There is possibility that those antibodies are not good for some specific population, but those antibody can be helpful for different population. So it all depends on the population. So explore clinical trial antibodies also. So here is an example of monoclonal antibody with malaria. So what happens if there is, so this is a life cycle. What happens when there is a mosquito, Anopheles? We all know that Anopheles, whenever it bites, it injects some sporozoites of falciparum and it enters inside the hepatic cells. And from liver, it runs to blood vessels where RBCs are present and they replicate in RBCs. One, it replicates inside the RBC, it forms merozoites and it ruptures and it goes to infect others. At the same time, if it's entered into gametocytes stage, the second anopheles, when it takes a meal, it will also go to the that mosquito also. Then it will infect other person. So this cycle is keep going on and on. So what happens? Some of the scientists are developing gametocyte monoclonal antibody so that they can inhibit this stage. Some are developing monoclonal antibodies for this initial stage. Some are developing for blood stage and some for hepatic cells. So if you are trying to develop some early stage antibody that could be from the antigens that are present during sporozoites. So in parasites, it's very difficult to identify that at which stage which protein is going to express. So there are possibilities that if the protein expressed on sporozoite stage, it will not be expressed on the surface during blood stage or gametocytic stage. So you have to be very careful during designing of your monoclonal antibody, in selection of your target protein. So this is one part. You can you can develop monoclonal antibody for inhibition of transmission. You can also, like, there is very complications during malaria. If if you are suffering from malaria, the patient has different complications. Like sometimes what happens, these RBCs, infected RBCs, coagulates, and sometimes these interacts with CD36 and into these walls. And this is a very complicated step, or you can say it's very complicated stage. If you don't avoid this CD36 binding of this infected RBCs, um, it will increase, like it will become a severe malaria. It will not expose to the immune system, and it will replicate and replicate more and more. So for that, scientists are trying to develop some CD36 antibody so that they can inhibit this kind of clogging or this kind of rosetta formation. So there are lots of way you can design your monoclonal antibodies. Here, transmission blocking. Here, rosetta inhibition. So what's happening in clinical studies? What we are doing in clinical studies, there are different um cases, or you can say different categories. So what happens? You can select some people that are previously exposed to malaria. What happens if you select some previously exposed person? There is possibility that they have some antibodies that could be a functional antibody that can control Plasmodium falciparum. Primary infected means if a person who is not previously infected, it's first time infected. So there is possibility that they don't have those antibodies, or they have very low amount of those antibodies against that falciparum. And if you took a healthy control, like they don't have any previous history of malaria, when you take the blood from all these three different humans, what happens? You can find very good antibody from this, like who have previously exposed, and you will have very small amount of antibodies from this, but you don't get any uh, antibody from this healthy control. But most of the time, if you are familiar with ELISA, with antigen and antibody perspective, there is possibility that this healthy control and also have some antibodies from other. For example, he has previous history of Zika virus. There is possibility that Zika virus antibody can bind the antigen of Plasmodium falciparum. So in that case, we can't say okay, the antibody present in this human also have antibody against malaria. No, that is just a cross reactivity. So for that purpose, we just remove whatever value we are getting from this healthy control. And there are some false binding also. Whenever there is antibody, there is always a noise that is known as false antibody or antigen interaction. Like they're not specific, the non-specific binding. So we have to remove whatever readings we are getting from healthy controls. Now again, there is possibility that you get a lot of reading during your ELISA for a specific antigen, but that is not uh, explaining that this antibody is functional. You have to study what are the history of this patient, if it's curing or not. If you got a very good antigen antibody interaction, you get very high level of antibody, it doesn't mean this is going to be a success. What matters if you have a very low induction of antibody, but it's a functional antibody, that is a successful antibody. If your antibody is not functional, no matter how much your body is producing that antibody, it's useless. So this is the basic of clinical trials, what happening in clinical studies, clinical experiments. So if you are planning to design some monoclonal antibodies, you should know about these basic things so that you can move forward with whatever you are designing. So if you are going to design monoclonal antibody using bioinformatics approach, you should know about these basic things so that you don't end up with some experimental failures. If you are not familiar with these things, no matter how good your antibody, you will fail in experimental. So this is basic from this study design. Now recombinant antigen protein expression. After designing of your monoclonal antibody, once you are done with your computational work, you have to express your antibody. For that, you can use phase method or you can use the traditional method that we use for plasmid expression, or you can use phase mid, whatever you are interested. So before going for expression or synthetic cloning, you should use SnapGene tool. There is a software SnapGene that you can use to check if your antibody is going to express in that cell or not, or your cloning is going to successful or not. So you can use SnapGene. And after production of your antibody, you have to purify. Of course, you have to purify your antibodies. After purification, you can use this method, ELISA. So what you can do, you can just use your antibodies directly from cells and you can check if it's working or not, or you can use some antibodies or tagged method, whatever you are like, whatever you have facilities, like it's basically depends on what are the facilities present in your lab. Now let's discuss about monoclonal antibody therapy, how it works. Suppose you have developed your monoclonal antibody. Here you expressed, you purified, and you have uh, validated that okay, whatever you have designed is already expressing if it's working or not. So if it's working, then what is the use? How it works? So here a simple example. This is a SARS-CoV-2 and the surface protein is in red color. So this green is your antibody. Whatever this green is here, this is your designed antibody. Whatever you have designed and expressed. What happens when you give your monoclonal antibody to a patient? Your monoclonal antibody is going to bind to this SARS-CoV-2 and it will inhibit the interaction of this SARS-CoV-2 to the host receptor. We all know that SARS-CoV-2 spike protein binds to ACE2 protein of host cell. So this is the ACE2 receptor. So your antibody is working very fine here. What is next? What other uh, field of antibody you can target? So here you can also target tumor cells or cancerous cells. What happens? Your antibody can bind to this tumor cell. It coat your tumor cells or cancerous cells, and it activates cytotoxicity by activating some perforins or enzymes from NK cells. If your coated tumor cell interacts with NK cells or the receptor FCR, what happening? They release some perforin enzyme that results in tumor cell lysis. But if uh, this tumor cell is interacting with macrophage, this is going to initiate the phagocytosis. And during phagocytosis, they will kill this cell. So either you can target some virus, pathogens, or in infectious disease, or you can target tumor cell or cancer. Now let's talk about conjugated antibody. So here antibody drug conjugate are hybrid therapeutic that combine the specificity of monoclonal antibody with the potent cytotoxicity of small molecule drugs. So here in the purple color, you can see this is your immunoglobulin or antibody, and this red star-like structure, these are drugs. And the black region here you see thread-like black structure, this is the linker. So antibody drug conjugates are designed for targeted delivery, as I told you in previous slide, that we use for targeted delivery of drugs for cytotoxicity or minimize the off-target effects. Like suppose you are giving 10 mg of drugs to a patient, cancer patient, it's not working. But if you use this antibody targeted if you use this antibody targeted therapy, what happens? You just use only 5 microgram of your drug, conjugate with antibody, and it will work very fine. Because whenever we take drug inside our body, it dilute during our dilute inside our body system. Like there is blood and some other fluids inside our body. So it neutralize, or you can say dilute. It's dilute your drug, and it's also uh, there is possibility that this drug will not be able to go to that specific cancerous cells. But if you are using this antibody conjugated drugs, that will directly go to your targets. For example, a person is having a hepatic cancer. So you need your drugs to go to the hepatic cells. There is no need to transfer your drugs to all the body parts. It's a waste of drugs. Even in a small quantity of drug, you can just treat that patient. So you just use some antibodies that are against some hepatic antigens only that are present on hepatic cells. And then these antibodies can bind to only hepatic cells, and they will provide these drugs for further process. So these are some other cancer therapies of monoclonal antibody. You see, they sometimes they do signaling pathway blocking. What happens? We all are familiar with EGFR. I hope all are familiar with EGFR. So these EGFR, when interacts with ligand, it activates some pathways, and during activation of this pathway, it helps to these cancerous cell to proliferate more and more. But if you are using some antibodies like cetuximab or panitumab, what happens? They stop the interaction of this ligand to this EGFR receptor. So if this ligand is not interacting with the EGFR, this signaling pathway will not activate, and this will stop the growth of cancer cell. This is the first approach. Second approach, what you can do, you can design some monoclonal antibody that can interact with this CD19, and it can also interact with some NK cell receptors. During this process, NK cell release some granzyme or perforin that helps in lysis of this cancer cell. The third approach is complement dependent cytotoxicity. You can design some monoclonal antibody that can bind to some receptors and that can also bind with C1q that results to cancerous cell lysis. Another approach is ADCP, that is antibody dependent cellular cytotoxicity. So here you see, if it's tafasitamab, this antibody can bind with CD19 and it also binds to the receptor of macrophage. So this macrophage is going to interact with this cancerous cell and will destroy. Now you have 4 different categories. You can design your monoclonal antibody either signaling pathway blocking. So your monoclonal antibody could be a signaling pathway blocking, or it could be targeting ADCC. It could be complement dependent cytotoxicity, or it could be macrophage based. So you have to decide which category you want to work. Now let's talk about computational approach. Because without getting the knowledge of what is happening in the wet lab, you can't start with computational approach. Now let's talk about computational approach. So in recent years, computational and AI-based methodology for antibody development are becoming more important. And NGS approach, affinity prediction, antibody designing, machine learning, B-cell apto prediction, deep learning, immunogenicity, and MD simulation are major parts. AB Diver is a tool that employs the publicly curated B-cell receptors, NGS datasets, and compares design sequences to natural reporters. Another is RAPSIC dataset analysis platform with an integrated antibody database that is also known as RAPID. AB Diver is a tool that employs publicly curated B-cell receptor, next genome sequencing datasets, and compares design sequence to natural reporters. So RAPSIC data analysis platform with an integrated antibody database is known as RAPID. There is another database that is known as structural antibody database. This is the link for that database. Another one is therapeutic structural antibody database. There are CADD, TAB. And another one is TPB2. It's also predicting some structures that is Rosetta based. So in this deep AV tool, you have to log in, and after login, you can easily work with this. Nowadays, you can also use some AlphaFold method, not only Rosetta based, but also you can use AlphaFold method. So if you're going to design your monoclonal antibody, you have to keep these things in your mind. The two main important thing is paratope and epitope. So the antigen part that is interacting with the antibody is known as epitope. And antibody part that is interacting to this epitope is known as paratope. Always keep these two things in your mind. If you're going to design antibody, there is some epitope and there is some paratope. So what is happening with this paratope? You see there are some CDR regions. Each has 3 CDR regions. Either it is light chain or either it is heavy chain. Both have 3 CDR regions. And every time a new antigen interacts with this paratope region, the CDR region will change. So if you are trying to design some antibody, this is going to be your target. You have to work on this CDR region mostly, because these are not like these are not our interest of regions. Like these are very constant regions, so we don't have to work on this part. We just have to work on this VL region or VH region, specifically these CDR regions. This possibility that all the structure of this immunoglobulin or antibody can be constant, but this CDR region changes every time there is changes epitope or antigen. Now, how many type of epitopes there? So in previous class, I've already told you that there are two different type of B cell epitopes. It could be linear B cell epitope or it could be discontinuous epitope. In case of linear epitope, there is continuous amino acid sequence. And in case of discontinuous epitope, there is conformational changes and two amino acids could be different from each other. So there is no continuous in this type of epitope. It could be single amino acid from different regions, different positions. Till now, I hope everything's clear. So we can start with our machine learning framework. So if you are interested in designing your own models, you can use this machine learning framework. What you have to do, the first step, if you are interested to design your antibody, you have to select your disease. It could be pathogenic, cancerous, or any other non-infectious disease like Alzheimer's. So right now there are lots of research ongoing on this Alzheimer part because scientists have found that there are certain proteins that are responsible for increment of Alzheimer. So what you can do, if you can design some antibodies that can bind to the specific protein, that will decrease the effect of Alzheimer, and it can also inhibit the further growth of Alzheimer. So this is a latest study. If you're interested, you can read more about this in literatures. And once you decide your disease, you have to select your target. Select your target, like which protein you are going to work or which pathway you are going to target. After that, you just have to design your monoclonal antibody. For designing, you can use deep and AB, SCV PRED, DNA star. So DNA star is a you have to purchase, like it's very expensive. So if your institute or your research center has facility to purchase this DNA star software, you can go with DNA star or Schrodinger. Or if you don't have that budget, you can go with this SAB Trade. This is freely available online tool for designing of your monoclonal antibody. You can use this tool. And MD Sam, can anybody tell me you have this MD Samuelson classes yet or not? Do you have classes for MD Samuelson already, or you are waiting, like in future you are going to take MD Samuelson classes?
We have in
No, they have
food
done.
animals.
They have done.
Okay.
They have done.
Then you are ready to design your monoclonal antibody. Design your monoclonal antibody, and you can use this MD Samuelson and docking. Of course, if you are familiar with MD Samuelson, you are familiar with docking. Once you design your monoclonal antibody, dock your monoclonal antibody with your specific antigen, like target antigen, and then perform MD simulation. If it's showing very good response, then you can move forward. Now there is another software known as AgriScan. So once you are done with your simulation part, you have to check if your designed monoclonal antibody is able to work in biological system or not. For that, you have to check the solubility and what kind of their surface accessibility, their negative charge, positive charge. So you have to predict those features by using some tools. You can use machine learning approach, or you can use freely software. Like there are some online tools, you can explore those. And if you're planning for machine learning model, like most of the group try to design some models because it's also very trending. Machine learning, AI, they are very trending nowadays. So if you are planning to work on these topics, it will be highly demanding, and you will get good publications for machine learning and AI based topics. After this, you can you can design linear regression models or SVR models. And after that, you can also cross validate. So this is the overall framework. Now come to the original research or protocol. Suppose you are planning for development of a model that can help you to design your own monoclonal antibody. In that case, what you have to do, you just have to download antibody sequences. And we all know that antibody has heavy chain and light chain. Just download those both of the chain, and you can generate some sequencing coding. Sorry. And then you can generate some matrix. You can generate nodes, edges. Based on that nodes and edges, you can generate graph transformer. You can also generate some multiplicity. There are different properties you can include. You can include CDR regions specifically, or you can just use whole region. But I suggest instead of going with the whole region, go with CDR regions, because working on CDR region is very specific and it's highly successful for uh, a, a, a good monoclonal antibody designing. So if you're targeting CDR regions, it will be very beneficial for your work. It will also reduce the risk of failure in wet lab experiments. So always try to download the CDR regions of as much as either it is in clinical trial. Just download the CDR region of clinical trial antibodies also. Now you have all the data. You can use template structures, like there are PDB database where you can easily download the structure of antibodies. You can use this structure based, and you can again, now there is sequence based. Now you can use also template structures. This template structure will help you to design your own models that can be used for structure prediction. By combining both of the thing, like CDR based approach and then your tertiary structure template based model, when you combine both together, what happens? Based on the scoring of metrics of this CDR based region and the structure based region, you will be able to generate your monoclonal antibodies. It could be Rosetta refined based, or you can use AlphaFold. So AlphaFold, you have to log in and you can ask for 1 month free trial for AlphaFold. It's yeah, it will take only like 2 weeks. 2 weeks is enough. So you can use AlphaFold also. So this is the overall view. If you are interested, you can design some model prediction or model designing by using this approach. Now, the second approach is you can just go to the PDB database, just directly download all the antibody antigen complexes. So there are two different kind of structure present on that PDB. One, only antigen structure. Second, only antibody structure. And the third is complex of antigen and antibody. So what you have to do, you can use two different approach. One, separate download structure, and second is antigen antibody complex. So what happens when you use this complex structure? The metrics will be different. And if you're using separate, uh, if you're using separate antigen and separate antibody, the metrics will be different. Why? Can anybody tell me why? Because whenever antigen or antibody interacts each other, there is some conformational changes. And it's not only about antigen antibody. Any two proteins, whenever they comes in the interactions, there is some kind of conformational changes. And because of that, the matrix will change. Make sure don't mix both the data together during designing of your model. Always design models separately. So this is an example. What is happening here? This is just a SARS-CoV-2 pathogen. You can see this is the RBD region of spike protein. So the red one is spike protein and this specific region is RBD region. This RBD region is going to interact with ACE2 protein of human. So if you go to the PDB, you can easily find there are lots of complex structures of ACE2N antibodies and lots of lots of um, antibodies that are interacting with RBD. So in that case, what you can do, you can just download the structure, and the second part, you can download the sequence of CDR regions. When you download both the regions, so if you are talking about RBD, it could be epitope or paratope. Can anybody tell me? If you are taking sequence from RBD, it will be paratope or epitope.
Parrot.
Very good. So it's okay. No. Sorry. If it's RBD is an antigen. Okay. So
Epitope.
RBD is
Then
antigen. So
epitope.
it's epitope. Okay.
Okay, ma'am.
And if it's antibody structure, the sequence in the antibody structure, that is paratope. Okay. So here RBD is antigen. So whatever sequence you are getting on RBD, it should be epitope because RBD is our antigen. And whatever sequence you get from antibodies, that is going to be a paratope. So if you are designing your antibody, you have to design paratopes. For paratope, you have to work on the sequences. So every sequence has some kind of motifs. Whatever this epitope and paratope, they have some kind of motif. Suppose this is the epitope. Not all the amino acids are going to interact with antibody. What is happening here? V and N is going to interact with your antibody. So this is known as motif. Similarly, in this case, you see D is in red color. D is the motif here. And here you see Q is the motif. So this region is multiple sequence alignment of CDR region, or you can say epitope region of this RBD. And these are the motifs. So motifs is very key factor of designing any kind of model. So this is sequence based. Now the structure based. In structure based, you use some kind of coordinates, like at which um, binding pocket it's going to bind. So at which level we have to design our antibody that antigen can interact. So we have to work on pockets. So this is structure based method. So once you combine all these together, what happens? It will generate some kind of genetic algorithm, or it will help you to select variants. It will also help you to select crossover and mutate. And at the end of this all process, you will have some kind of graph that will tell you the success of your model. It could be AUC based model, or it could be can say specificity or probability based. So what kind of studies you are using, what kind of model you are using, it depends on that. So this is just an example. If you see here sequence logo, I hope if you are working on uh, proteomics data or genomics data, you all are familiar with seq logo. What is happening if you use thousands of epitopes or thousands of paratopes, you will get some kind of motifs or some seq logo. Based on this seq logo, you can identify the motif, and this will help you to generate your model so that you can easily identify, okay, if we are going to design one antibody against SARS-CoV-2, specifically this RBD, there should be some W present on this 463 position here. And here you see the E at 484 position. So these two could be the major, uh, it's just an example in case of RBD. These two could be our major motifs. So based on that, if you design some antibody, there is high possibility that that will go to interact with your RBDs. Now this is just an example for, like, we designed some monoclonal antibody for SARS-CoV-2 spike protein, and this is the interacting residues. You can see the yellow region, and you see there are 4 epitopes, and epitopes are in also different colors. You can explore. I hope all have familiar with PyMol. So when you go to the PyMol, you can easily visualize this heavy chain, light chain, and whatever the antibodies. These are the references. If you are interested to read more about these things, you can read these references. Any questions till now? Okay, if there's no questions, I will move forward with demo session. So here I'm taking example for malaria antigen AMA. AMA is an antigen for malaria, and this is the PDB ID 2Q8A. So this is one example that you can see here, 1, 2 cubes, 8 cubes. This is the PDB ID. So you have two options. You can just go to PDB and you can download the PDB file, or you can go to NCBI. On NCBI, there is also options like you can just paste this PDB ID, and if you search, you see there is heavy chain, light chain. You can get the sequences from here. I hope everyone is familiar with NCBI and PDB. So I'm just showing so that you can easily download these sequences from NCBI or PDB. In PDB also there is some options. You can also download FASTA sequence here directly. So I'm just showing what else you can do. Like if you want to learn more about these proteins, you can go to the NCBI or you can use PDB. So this is just for sequence retrieval. How to retrieve your sequence. You can use NCBI or PDB. Now let's talk about the tools. So there is a tool A Body Builder ML that is from SAB Pred. You can use this tool for designing of your monoclonal antibody. Another one is AgriScan 3D. You can also use this directly uploading your PDB files, or you can write your PDB code. Now the next is B-cell epitope prediction. You can use ABC Pred, or you can use IDB tool for B-cell epitope prediction. If you want to predict your antibodies, you can use SAP tool. This is the tool that you can do. Here there are lots of options you can explore. You can search for CDRs, you can search for structures, you can search for sequences. Now, if you want to predict the structure, you just have to paste your heavy chain here, light chain here, and you can give any file name. For example, if we click heavy chain, so always remember heavy chain should go into the heavy chain box. and the light chain should be inside this light chain box. Now, if you start the model, so it started, you see, it started, and this is machine learning based approach. So here you can see what's going on. They are using some templates. They are searching for templates for these specific sequences. It will take 2 minutes. So we can just, okay, we can go to PDB here. So here you see this PDB contains AMA antigen. It also contains light chain and heavy chain. So this complex has 3 structures. So suppose you have this antigen and you want to design antibodies. What you can do? This is your antigen. So there is possibility that there are certain epitopes that are interacting with this antigen. So there are some epitopes and there are some paratopes. So whatever amino acids present on this amino acids, there is possibility that they are going to interact with the paratope of antibody. So what you can do, you can just download this structure and open in PyMol here. Or whatever tool you have to visualize your tertiary structure. If you have Discovery Studio, you can use Discovery Studio. And if you go to the chain, you can also visualize that there are 3 different structures: heavy chain, light chain, and the antigen. So here you can see there are dotted yellow lines. These dotted yellow lines are so in these dotted lines, you can easily target your. So if you have Discovery Studio, it will be easier to work. Just find out whatever interacting residues are here. And by using these interacting residues, you can prepare some list. That is what kind of epitopes present on this antigen, and what kind of paratope present on this antibodies. Once you prepare those lists, okay, here it's finished. See, this is your design monoclonal antibody. One is heavy chain, another is light chain. The blue one is heavy chain and this pink one is light chain. So here, you look at the heavy chain. You see this blue line? This blue line is CDR region. So every heavy chain and every light chain, okay. So